Greetings, everyone. On behalf of the GeneBank platform and the Crop Trust, welcome. Glad you could join us today. I'm Yanni Van Veen, and with me is Faye Wambua Ludeling, who is co-moderator for this presentation. I'm very pleased to introduce our speaker, who is at the crossroads of several distinct fields, in particular computer science, molecular science, and the visualization of the omics as they apply to agriculture. She is here today to show us how not to drown in our own data. Dr. Trupti Joshi is an assistant professor in the Department of Health Management and Informatics at the University of Missouri, Columbia. Her expertise is in the fields of bioinformatics and its application to plant sciences, animal sciences, biomedical sciences, and health informatics fields. She has published more than 127 scientific papers and co-developed several frameworks for genomics and multi-omics data integration in the agricultural and biomedical domains, and the development of computational methodologies and data analysis pipelines using cloud-based resources. The title of her presentation today is Soybean Knowledge Base and Knowledge Base Commons, Enabling Data Mining, Farming, and Harvesting in the Cloud. Welcome, Trupti.
Thank you very much, Yanni. Good morning, good afternoon, and good evening to everyone joining from all over the world. This is truly exciting. Really wanted to thank Cropsus for giving us the opportunity to share our development expert and experiences. I was very excited when Yanni reached out and to learn about what the CGIAR is trying to achieve in terms of the one CGIAR platform and the challenges that pretty much everyone is facing with the data integration. And I think this is something where I can certainly share a couple of our experiences and introduce you to a few tools which you may find interesting, some that may be directly applicable, some others which might kind of spawn some ideas in terms of what can be done to do a better job of these integration pieces. Definitely, I think this is very exciting opportunity. I always enjoy when we get a chance to sort of, work more closely and interact with our user base. I hope this sort of gives you an idea in terms of the pieces that we have developed. So I definitely would like to kind of start by, taking you through our journey in this whole infrastructure developmental pieces and the tools that we work on to address our real world problems. In doing so, I kind of have a personal passion of trying to work on these translational and applied pieces, which impact all domains of life, right? Not just plant sciences and agriculture, but also biomedicine. If you're working on kind of public health side or animal science. And so it kind of, this pattern stems from my prior background in biomedicine, as well as bioinformatics. And I kind of, like to mesh these together, like to marry these fields together, where we can get all of our computational pieces tied very well to the wet lab or to the field side. And that is essentially what I like to take you through, just to see what all is feasible and can really be achieved in terms of computational and bioinformatics pieces. I'd like to go ahead and get started. Thank you, Annie, for introducing me. I am in the School of Medicine, but I also do a lot of interdisciplinary collaborative work with our informatics and data science program, which is MUIDSI, engineering, our interdisciplinary plants group, and then life science center. And so this kind of, as I go through that, I have tried to put a lot of examples which are more related to crop on the agriculture side, but you may occasionally see a couple of places where I will point to a few details from maybe the biomedical side, which kind of also helps us, in terms of when we are building these pieces. So I would like to start with a quick introduction and then move on to touching upon a few of the developmental and all publicly available tools and resources that we have built on. And so you'll kind of see me flip through a couple of slides pretty quickly, but to give you an idea of how the flow goes. But I will be very happy to kind of answer any questions that we have towards the end. So I think everyone here is already feeling the change that next generation sequencing technologies have really brought. In the last about decade, a little over a decade, they have transformed research considerably, and now it is feasible for us to be doing whole genome scale and multi-omic studies on those with several different crops or other organisms. And this really has led through this plethora of data. What we are seeing here is really only from 2014, and you can already see how the spectrum has been continuing to increase, whether you're looking at targeted resequencing or sequencing data sets, whole exome sequencing, whole genome sequencing. And this is really true in all domains of life, whether you're working on a clinical side, whether you're working on the agriculture side. So all of these NGS technologies and high throughput technologies impact all living organisms. And so we kind of work and we have pretty much touched most of the domains, whether you're working on viruses or bacteria, various animals or model organisms. At the same time, we have done a lot of work on different crops and on the biomedical side. So, what this really brings us is a spectrum for us where we are generating data sets at all of these different levels, and they are really telling us everything that's happening in that plant or in that crop at a given time. So, you could be looking at your genome level information where you have your genome sequence, resequencing, you know what variations exist in your different plant accessions or plant introduction lines, how methylation changes and chromatin changes might be impacting them. The idea is to sort of integrate them together with the transcriptomics so that we have an idea about the gene expression profiles, the microRNAs or other small RNAs, and how they interact with each other in terms of co-expression. Then the next level is the proteomics level, the expression of the proteins, their localization, their interaction with others in terms of protein-protein interactions or interactomes. There are a lot of studies being done on the metabolomics level where you are measuring these profiles as well as studying hormones at the same time. And then still studying the mutant and the natural variations that exist and have really a need to integrate all of this together so that we are better positioned to make new discoveries or decide what needs to happen in certain environments as we continue to advance our crops and other cultivars. So bioinformatics and computational biology has really become an essential component of all of these domains, and we kind of have been touching and working with them. What's really challenging? People who have been doing this for several years, but what has really changed is the amount of data that gets generated from a sequencer every single day, something that could have been done or needed almost a year. And then the diverse nature of these biological data sets, that these all have to be integrated together. You're not comparing apples to apples all the time. You have to have some kind of a translation happening. And then how do we convert all of this data into knowledge and make really meaningful discoveries? So what I do hear most commonly from all of our collaborators who are more on the field or the wet biology side is this whole concept of drowning in the data, right? It's just too much. It's just too much for anyone to be handling and looking at beyond, Excels. And so really there is the very much need of finding infrastructures would make it a lot efficient and easier to navigate through these data sets. So the multi-omics data, another challenge that we face is that if you want to tap into something that exists, has been published and deposited in any databases, they are kind of scattered in different databases. And so what we really, our users find difficult and challenging is how do you bring it all together? How do you pull it all together in one place and not have to go and work with several different resources? Now, this is very true in, the field of precision medicine, which is where my primary home department is. We face with similar challenges in trying to predict, diseases and what are the genes or the mutations that are linked to rare diseases. How do we design better drugs and which patients might be better suited for treatment with a certain drug based on their genomic variations? How do you get a better understanding of patients who might respond optimally to these drugs without having side effects? Or how do you really build amazing, biomedical platforms which can allow you to tap into entire clinical data sets, but not miss out on getting all of this genomic information? The same is true whether you're really thinking about crop sciences and agriculture with the goal of food security. We know the world population is projected to hit about over 9 billion by the year 2050, and that's really going to require a global increase in various crop productions that needs to be at least doubled by that time. And so this is a graph which shows that in terms of the solid line as to where we might be going with the current pace, but where we really need to be with the dash lines projected for four of these major crops. Now, boosting crop yields is possible if you can increase your cultivating land, but that is really shrinking day by day. And so, how do we really boost the yields? And can we use or make use of informatics to really tap into or get a better understanding of the crop germplasm and the genomic information tied with it, so that we can do a targeted breeding and select better cultivars and accessions to really move forward, and the ones that might be most suited for certain locations. So everyone working in this field, I think, already has a pretty good sense of how and where agriculture is headed. There's a big focus on modern farming to really apply more and more techniques based on available data or information to go towards precision agriculture. And so a lot of these decisions are made based on the agronomic data sets. And so you're seeing here fields, but then you're seeing here fields where there are certain crops being grown in certain areas, or there are information about the soil quality and so forth. And so there's a lot of stress on selecting hybrid or plant populations, or selecting the genotypes to be utilizing based on what is the yield that is predicted or anticipated, and then how is the soil data looking like, what sort of pesticides, fungicides have to be applied to get these optimal environments, and then how does the economic market prices and everything flow into that. So there's a whole cascade and a whole circle of information that is available, and so all of this really can be taken into account through infrastructures that allow you to tap into these different pieces, and then use them towards applying or understanding, various crop diseases and making decisions about environments suitable for growing certain crops. What we do see is that the model has really changed over time. And so we are used, maybe used to the older way, where a few organizations used to generate these data and others used to consume. If you think about it more in terms of, a few news outlets, or maybe the newspaper, which is kind of becoming, almost obsolete maybe these days. But the new model really is where all of us are generating data and all of us are consuming data simultaneously. And so a good example of this, I think, whether you like it or not, would be Facebook, Twitter, any kind of social media, any kind of blogs. And you have these as different information sources. And so of course you have a lot of data generated, but that at the same time, some of it might be relevant, some of it might be irrelevant, and so you really also need to know how to weed out and differentiate between those. Now, this has really started impacting modern day farming. I think, though I wouldn't say these are, the gone days, this is how you would think of as in terms of a traditional farm. I think what is really happening is there's a lot more technology that you get to see in farms these days. And so a lot of drones and so forth are also applied in terms of trying to understand the phenotypic or gather the phenotypic information from imaging and so forth at the same time, in terms of where you're measuring genomics data. And so you can think of it that there are now several pieces of information which are data rich, and kind of have a more need of doing computational integration as compared to what existed in our traditional farms. And in order to really deal with that, we have to think in terms of modern day farming and agriculture being really as much about data as it is about digging and harvesting, because only then are we going to position ourselves to be looking at and building infrastructures which can better deal and better address the challenges which all of these different data sets bring for us. So I think you may have probably already heard a lot of these terminologies, but there's definitely a data explosion on the agriculture side as well. A lot of terms have been emerging and are tossed around in terms of data analytics, data mining, harvesting, data science, integration, and this is only a top of the list. So many times there might be questions in terms of what is really applicable. Do I need to do all of that? Is there overlap between them, or what is really relevant for the pieces that I work on? And there is no exaggeration that this has really led to a transformation of the way people think about information, and they build systems to collect it and analyze it. And so earlier, maybe the most common word would have been databases, but as the information grows, there has been a very large focus on other pieces, other terms involving data, if I may say so, such as data repositories, data marts, data warehouses, data lakes. That's kind of the latest one. It just kind of keeps growing. But some of these could have structured data, some of these could have raw data sets or unstructured, and then you really go and fetch. The sizes would differ. You would really go and fetch the information that you need based on what you're doing at that point. The most common pieces that I would say is data mining process, where even we do a lot of work that I will show you in the next few slides, where we deal with a lot of these decisions, right? We are looking at digging through the information or the data that's already collected with patterns, correlations, and trends. All of that sort of encompassed in data mining, where we are trying to apply computational methods like machine learning, deep learning, build infrastructure or databases, frameworks, maybe apply some statistics, mathematical modelings, or try to look at patterns and make inferences based on that. So that is still the most common application that I would say. Data farming, where at times you have the need to grow or acquire more data in order to do a better forecasting or modeling. So you might be doing some targeted experiments or targeted studies in different locations, or at times even computationally growing the data so that you have enough power to apply some of these computational models. And so a little bit less, I would say, along these lines. And then data harvesting. And so these kind of, I mean, harvesting might look like, oh, maybe that's most relevant to our piece, right? But this kind of goes a little bit beyond data mining. It's very similar to that, but the key difference being that they extract and analyze data collected from online resources. So there might be things like publications, articles, blogs, and others, which might have very relevant research information. And so this is where a lot of web crawling or automation is done in terms of pulling this information. And it's something that really continues to grow or thought of as a renewable resource. And no matter which area you think applies, I think what is very clear is that big data is here in agriculture. And so I think the earlier and the better we look at infrastructure and systems which enable us to utilize them efficiently, the better we are going to position ourselves to solve the problems coming in future. And so you may be focusing on just pure integration or analytics, or you may have a need to, look at just one piece, which is infrastructures or databases, or how to manage them, how to store them, or perhaps maybe a visualization component, or multi-omics integration, or you really want to go into, data protection, privacy, or even apply much more advanced artificial intelligence tools. There's a whole bit of pieces, and this is a mesh of pretty much everything interrelated and correlated with each other. So what my group really focuses on is this enabling this translational research. As I said, we like to dive in deeper in the available and existing diverse nature of the data sets. We try to tap into knowledge that might be currently publicly available, such as gene functions, pathways, maybe network regulation information. We build algorithms and infrastructures to deal with that. And then we love to work on application side. And so given my background, we work on everything from precision agriculture, precision med, One Health, where we're trying to also bring links between animal and clinical side on the biomedicine, and then pretty much apply a lot of these to biomarker discoveries, or even, for that matter, studying viruses, through SARS-2, COVID, for example. So here I think I would like to switch gears and start giving you a kind of feel in terms of what are these infrastructures that we have built and how you might be able to take some pieces that might be relevant from it, or at least get a sense of what goes into building a complex system like this. So I'd like to start with soy kb. This is kind of what I call as my baby, because this is something that we started developing more than a decade ago, back in 2010. And the whole idea behind that was because no infrastructure existed at that time, and we were really looking at generating data on all of these different omics levels and had the need for integrating it. And so we really started thinking in terms of this one stop shop infrastructure framework where we wanted genomics, but we wanted to make sure we link it back to breeding and phenotypic data. So all of our users or all of our collaborators who really make decisions about it are able to utilize this information. We succeeded in linking it back to high performance computing and Cyverse, which kind of used to be iPlant. And so you may be familiar with one or the other name. And so we were able to link it back for computation or staging our data and pulling information, which is beyond like a database. It's a publicly available resource at soykb.org. It's very widely, published, and so you'll find information as mentioned in these papers. And then we still have like a combination of, registered URLs. You really do not need an account unless you want to do certain analytics. But then we have users from academy and industry and all over the world. And so most of the information on SoyKB is public already, since most of the data sets have already been published. I just want to kind of quickly give you an idea in terms of what visualization capacity we have. Something that looks like a heat map and a clustering. You might be looking at gene expression or protein expression data sets over here and pulling for a subset from and then different conditions. You can pretty much look at that same information on the pathway viewer, where you might throw in proteomics, transcriptome, or metabolome and look at which parts of the pathway might get highlighted. If you're interested in looking at protein structures, it has a visualization tool for it. Or if you want to look at, how your gene expression changes in the different tissues that you're looking at in a particular system or a particular condition, you can essentially use kind of tools like AFP browser, which put that data in terms of pictographical representation. The most common and a very well utilized tool is this differential expression suite of tool for transcriptomics data. The same thing works for proteomics as well. You can decide which data sets you want to integrate together or select and do a comparison. You can do 4 at a time. And so you'll see here, it gives you in a Venn diagram an overlap between things that might be common or uniquely changing in each of these comparisons. And you can simply click and get access to the list of genes which are differentially expressed, plot them onto having an idea about functional annotation information, or really see which of them might be upregulated, downregulated, and so forth. So you can make decisions about which genes to focus on for your follow or downstream studies. In doing this, we have over the decade, piecemeal, one piece at a time, depending how our priorities with our funded projects aligned. We have built a lot of tools in SoyKB, and so definitely, in the interest of time, I won't have a chance to go over or touch upon a lot of them. But this sort of also will give you an idea about the visualization pieces, all the data sets behind in the MySQL database, and then gets pulled in terms of what the user is querying online. And so feel free to look at the publications, and that will actually give you a lot more in-depth information about the capacities that exist. From our experiences from SoyKB, and as I had, through our journey, getting more and more involved with other crops and research teams through our NSF funded projects, we started thinking and we noticed that there is a very similar need beyond soybean for a lot of other plants and crops, maize, Arabidopsis to begin with. Through some of our collaborations with med school, we had similar needs in terms of, studying biomedical diseases. And so what really we decided to focus on is building this KB Commons informatics framework, which is, instead of having to go and reinvent the wheel every time for a different organism, why not centralize everything so that you can have a comprehensive system which can deal with any organism, no matter what, as long as the data formats and the data going into that fall under genomics and multi-omics domain. So we focused on KB Common System, which now addresses a bigger challenge with storage, management, sharing, doing data integration and visualization for the entire multi-omic spectrum. We borrowed ideas from Soy KB and pieces that were relevant and innovated on the other pieces. And then we still connected to high performance computing, but now actually have different other resources that you can go to and have been able to enable analytics and so forth in it. So for the KB Commons, we went beyond that to using Laravel framework, to an expandable framework, and then more in terms of JavaScript and expanded with the database as well. So anyone interested in kind of the technical details, this should give you a lot more information about the infrastructure piece. We built it in such that it has 4 different modules, a database module, a processing module, because now we want to give our users a lot more functionality to do things on their own instead of having to come back to the development team each time. So we also have the data access module, which talks between the database and then the web interface, because this will now support multiple crops and organisms. And so the functionalities are split between these 4 different modules. If you go to the KB Commons homepage, which is accessible publicly at this URL, you will see you can essentially pick and choose which area you want. And then I'll touch upon the 4 new functionalities that we built in KB Commons, which do not exist in Soy KB. Again, there's this has been widely published, and so that will give you a lot more details. The KB Common 4 pieces, the main thing that differentiates it from Soy KB is that any user can create a new knowledge base here. So whether you're working on another, like a legume, or maybe something like brassica, you can really go and bring your own or build your own KB. You can contribute multi-omics to existing KBs or the new KB that you build. You can update your genome versions on your own without even having to come back to the development team. And then if you just want to simply go and browse, you can do that. You can pretty much do all 4 of these things if you're starting with a new organism and bringing your own data sets. And in doing so, we have actually, this is an example of a couple of things that we have already worked on and that exist in the KB Commons system through all of these 4 different species groups, I would say. So I will quickly flip through a couple of steps. If you want to create a new KB, you can start by selecting which organism, which version you want to work with. It will tell you to bring 6 essential files, which are mainly going to be tied to your genome sequence, your genome sequences, and then their function annotations and so forth. And then once you bring them, they are in the correct format. The system will check for it and then allow you to upload them, and then you're good to go. Your basic structure of your KB is essentially established. So either you can go and browse that information, or you can go to the next step where you want to build and add more multi-omics data. So adding multi-omics data, again, you can either start with that earlier step or select a organism and version. And the version is important because a lot of the things like gene models or SNPs are going to be tied to that certain version of the genome sequence. So we always allow you to do that selection first. You can then decide if you want to bring transcriptome, microarray, RNA-seq, proteomics, pretty much any other, methylation, epigenome. You select how many data sets or how many differential comparisons you want to bring. And then the system will guide you through next processes so that it will allow you to upload files, and then it will incorporate that, put it into the database, and then make it available for you so you can browse them later. For updating genome versions, you have a step where you select the organism and then you just bring in your own, the gene models and then the genome updated FASTA sequences. We prefer to stick to phytosome for plants and ensemble for other organisms just because it's a very well established and a well tested approach. Ensemble these days, I think, is growing a lot more for plants as well. And so I think those are also formats that have started working pretty well with KB Commons. Once you're done with that, you can actually browse KB and use a lot of tools that have been already developed, and we continuously continue to add new developments here, and so you'll always see these kind of changing. a lot of data that you bring in could be private data that you want to keep, limited to yourself, and only release it to others after publication. And so everything gets controlled based on your login and your groups, and you can share your data within your group. So you can essentially establish something that works where you have 10 people in your group who have access to all the data that you're bringing in. And so you pretty much as a user have control on a lot of these features and functionalities. The other piece that we did not exist in SoyKB earlier is that we allow analytics. So if you want to do your own transcriptome analysis, like a gene expression from next-gen sequencing, or you want to call your own SNPs and indels for any line that you went ahead and did whole genome sequencing on, you can use our analytics pieces so that our analysis will run on Exceed or Cyber's infrastructure and our local computation at MU. All you have to do is bring in your files, tell us what analysis you want to do, and then it will run on our machines and give you access to all of the results, which you can again bring back to your KB Commons organism or KB that you might be working with. In doing so, we were also able to kind of tap a little bit more into engineering aspects where we could bring in on-time URB, which we think of as an automated suggestion platform, where you can customize your cloud resources needs so that if you're bringing 10 files or 10 conditions for analysis versus 100 conditions, and whether you have a need to have your analysis results tomorrow versus maybe a week from now, this system will actually allow you to decide where to run these analysis and infrastructures out of maybe the several infrastructure pieces that we have. And so this kind of automates that system based on the performance, cost, and then the urgency of the analysis. And so with our engineering collaborator, Prasad Kalyam, we were able to tap into those and have been able to publish work along these lines, testing the performance of these analytics workflows. This same KB Common system has actually been used for other diverse applications. As I mentioned, we have these cancer-related and disease-related pieces in it. We have been able to use that for the initial setup of a neuroscience-based KB, and then Maze KB, which I'll just touch upon very briefly, through where we are generating a lot of data sets through our NSF projects, and then some future applications that we are considering and bringing in cross species, because now we have multiple crops in the same KB. You could have same drought or other conditions you are studying, and so you are now in a position to do more of a cross species homology based comparison in that same infrastructure. In terms of the cancer, just want to quickly scan through that. We have established things for the patient so that you could look at patient data, how their cancer oncogenes might be expressing, how population, other patients with those cancers express, and how whether your patient fits in that same model or is kind of an outlier. Cell lines, for a lot of research done on cell lines and looking at copy number variations or drug information or looking at clustering based on their expression patterns. So there's a whole suite of things that's enabled there. Clinicians looking at individual, cancer patients or a subgroup of all patients with that cancer and seeing the distribution based on their age, sex, tumor classification, and so forth. And so you can imagine, as long as your data fits into your genomics, multi-omics, of course, every organism is going to have its own uniqueness and things you might have to tweak. But overall, in an infrastructure perspective, you can enable a lot of this together. Then Design Neuro, use that to link it with some neurons and some MRI, brain fMRI data sets, and link it to gene expression. And then our Maze KB, which, as I mentioned, we have data coming from two of our NSF projects with PI Bob Sharp and PI Paula Maxine. One studying drought nodal root growth, and the other one tapping into auxin for looking at its functional domains that exist with that. And so we generated a lot of transcriptome, proteome, metabolome, ChIP-seq, DAP-seq type of data sets from these two projects. And so the one I'll just show in the next two slides is where we had a lot of these well-watered versus drought conditions for nodal roots split into three zones, A, B, C, for this lab FR697 and inbred line and maize, which is essentially, performing better in terms of nodal root growth as compared to the reference B73. So we were able to take the differential express genes about 3,000 from these three comparisons, combined with 500 or so metabolites from these conditions, and bring them all together to get an understanding in terms of The two components which differentiate between the root zones ABC and between well watered and water stress, and how many genes and metabolites can directly come out which can explain this phenotypic differences in terms of nodal root growth measurements, water potentials and things like that, and how we can link them back such that you see here these component one and then the component two here in these yellow and blue boxes. The difference that you see here is this whole thing is drought condition and well watered and then A, B, C. And so the component two here is the one differentiate between the drought water deficit condition and the well watered in exactly opposite directions. And then the component one is what differentiates root region or root zone A from B and C, as you see over here. And then that lands up giving a combination of genes and metabolites, which we have been able to link back to specific phenotypic changes. I think with that, I'd like to move on to the next one. We did generate a lot of whole genome sequencing data, close to about 1,000 soybean lines as part of a USB funded project several years back. And so in doing so, we generated a SNP calling pipeline, where we were doing SNP and indel analysis. We published it with making the source code and everything available for this PGen workflow, and you can just directly go to SoyKB and run this on your own if you have a few, resequencing lines you want to analyze. But then the data that was generated out of that generated about some 20, 30 million SNPs. And so really it's impossible for anyone to query through that data unless there is a good web-based user utility. And so in doing that, we built this next generation sequencing, resequencing browser, which gives users access to everything from the raw data to their quality reports to actual SNP annotations, seeing the effects of the SNPs, or start looking into what regions were affected by copy number variations from these different data sets. And so this is again publicly available to look at information close to about 1,000 soybean genomes on SoyKB. What came following that is that we were more interested in looking at how to take all of this data and link it back to the phenotype so that you can make better decisions about which areas are showing differences in terms of alleles or which is important to tap into in terms of breeding. And in terms of that, we built the SnpWiz tool, an advanced version where you can decide what data sets you want to combine, select which genome versions or chromosome regions you want to focus on, select the SNP effects you want to focus on, whether you only want non-synonymous changes or those impacting coding genes and others, and also bring in phenotypic information if you already have for those lines or you have already done a GWAS and a p-value significance based on that, so that you can look at the lines, they cluster based on the region you have selected and the SNP information. They show you the similarities in the SNP, the chromosome, the gene that's overlapping, and then look at the amino acid changes that might be existing for that particular position. If you did have phenotypic data, you can bring phenotype type here and color them to up to like 10 different colors based on whether you have quantitative, qualitative phenotypes to bring in. And then if you had a GWAS, it will also add in the p-value. So it tells you these were the regions which came up as significant hits in your GWAS analysis, and it allows you to tap into the causative gene and the regions around it, which usually fall around the GWAS areas and not exactly the SNP that came out. And so this really allows you to go ahead and inspect some of that. All of this can be done online. If you wanted to download and save the results, it provides the results in these three different formats. And so these can be even downloaded for sharing or for further analysis on your own. Now, since we were dealing with a lot of this data, we did see that from time after that, there were other groups that were really generating similar resequencing data sets. And so we had an interest in going beyond that and incorporating data from these other groups. And so that's what led us to our next project, which is usually, really the most fresh piece of information we have been working on right now, which is the soybean allele catalog. And so this has really come through 3 years, funded project. And so this really allowed us to get into a deeper comprehensive understanding of soybean diversity allele. That was our interest, tapping into US soybean ancestors and release varieties. We wanted to build a framework that can allow us to expand these resequencing data sets and create a curated panel that we can always continue upgrading when new and new data becomes available. And idea is to find alternative alleles that might be available by looking at all of this data that can be brought back into, the breeding process and see whether that can have an impact with various different traits. And so, in doing this, we really brought in and have curated and worked with these, diverse panel of 775. You will see they come from multiple data sets here, and so it also kind of gives you a distribution of how many of these are elite land races and glycine soja lines, and what percentages they form out of the 775. So this is our curated panel right now, and we've already made advance from this to about 1066, which is our next panel that we will be looking at. In doing so, we build another, other pipelines for SNP analysis and building and merging the data that we need that needs to go into the soybean allele catalog web tool. And so there are two pipelines that go with that, and we're kind of working on a manuscript draft at this point, but this is available online. And so if you go to soybean allele catalog, you can either query based on a subset of genes that you want to investigate what their allele diversity looks like, or you can also bring in some of the plant introduction or accession numbers in combination to one gene that you want to investigate in a more detailed. So the first one, which multiple genes, is you're just looking at two genes I pulled out from chromosome 1. And what it really gives you is that it breaks down the data into how many of these allele combinations. So you have different allele or SNP positions here in the columns. And so it's really representing almost like a haplotype combination for you. And how many of these are, numbers here show how many of these are coming from certain sojas, cultivars, land races, what is the total, or how many of them might be North American cultivars. And what you see here is the red, blue, green colors are highlighted based on SNP effects. So if you have some missense variants, then you are having blue. If you have red, which is a stop grain or a frame shift, or anything that affects your splicing site between exon intron, acceptor donor positions, goes in green. And so there's a few other color codings that we have. And then you can directly browse data here, or you can download. If you click on any of these numbers here, it will tell you the exact accessions that fall into that group together for that particular line and the SNP information. And so you can see here, these were about some 14 land races, and it'll give you the exact name, accession number, and for the gene that you were investigating, countries that they came from. So you can tap and look at that information in much more depth. The second piece, which was on the right side, allows you to look at particular PI lines and genes and pretty much, pulls out the information on the same pieces. But what it allows you to do is a more straightforward comparison as compared to the earlier one. And many a times the way our collaborators go is they want to investigate certain traits, certain group of genes, and then tap into things more detailed based on whatever jivas they have done. And then so they go and use our right side of the tool. So this really has enabled us to tap into and look at alternative alleles. An example is related to protein trait. I think most of you might be well aware there's protein, regions on chromosome 15 and 20 that people look at, and so you really have this green showing a lot of higher oil, higher protein, lower oil regions, and then the red, which is lower protein and higher oil, which is in Williams 82. So we are able to see that in our tool very clearly. And then what we find here is this one frame shift for this particular PI line here, which can serve as a novel potentially defunct allele for breeding if you want to try and modulate this protein and oil, because they kind of have this inverse relation. And so a lot of idea behind this is that maybe some of the alleles have been lost, or only certain have been progressed through the domestication and the cultivars that are used in North American side. And so is there a room for bringing more diversity in, which might still allow you to preserve or do that conservation? So you can see that here, and the same piece of information can be pulled from SNP EFF, where you can see that particular PI line that I showed you earlier, and where you see this deletion occurring over here, whereas most of these other have Z, which have like some insertion at that position. And that really allows you to identify and use these tools in a complementary sense. So that's what will come out hopefully soon in a publication. And then I just very quickly want to go and just introduce these two tools, which are additional pieces of work that we have done. This is Impress, which allows you to really look at pathway resolution based on whatever or omics combinations you might have generated from experiments. And so it uses all of the KEC pathways in the backend and protein-protein interaction, has a detailed algorithm, which I won't go in at this point, and the details are in these published papers. But when we apply this to our maze nodal root system, you can see it pulls up 5 of these significant pathways for our root zone A, which is the tip of the root, which is showing most of the cell elongation and progressive, growth. And so you see the color coding in the boxes really tell you the pathways and the genes associated with that. And then the color inside the box really tells you whether they were downregulated or upregulated genes. And so we put it into this with the idea of doing some in silico hypothesis generation to identify some key connectors coming from the data, and not just focusing on function enrichment or differential gene expression. So this has really led us to a lot of good insights into the data with our collaborators. Tool is available online if anyone wants to go and run their own data. It'll walk you through some of these steps and give you, the results at the end. So I think this might be again something that's very relevant. And then another very fresh discovery is bringing deep learning into prediction. So we have a G2P deep web server which does quantitative phenotype prediction and genotype discovery by using SNP data. And this is the first framework which allows users to train their own deep learning models online and use saliency for identifying this. So it's again published in 2021 this year and available online at g2pdeep.org. And you are actually able to look at the significant SNPs that might come up. So it's almost, complementary to a GWAS type, but it will give you prediction of new lines based on the SNP information without having to generate your own phenotypic data. So we have soy and NAM dataset as an example in that development. And so I think this is something that will give you a much more clear idea in terms of how it might apply to certain phenotypic datasets and genotypic datasets that get generated on various crops. So I think with that, I hope I was able to kind of show you a whole journey of and a suite of things and tools that we have developed over the time, which allow us to, apply and for a lot of these research applications, to name, a lot of these frameworks and pipelines and web-based tools. So this is, I would say, still just the beginning. There is a whole lot of, artificial intelligence coming, which is kind of a step further from this, which I'm sure a lot of you have already heard about, and it is probably going to get even more integrated well as we progress through the future years. But I think that's really exciting because we do want to make a big leap and not really get drowned in all of this data, but be better positioned to be making a lot of new predictions. And so with that, I'd like to thank our team, a lot of graduate students, postdoc research scientists who have been working in development of these algorithms, tools, frameworks. Some of my key collaborators, a lot of brainstorming with Kristen and Maria on our soybean projects. Our funding sources, some of them on the maize and then engineering side, and our United Soybean Board for all of the work related to soybean, NIH for biomedical. And with that, I would just like you to point to our group's website if you are interested in looking at some of the other tools which I didn't get a chance to touch upon, the most two common frameworks that I talked about. And feel free to reach out if you have any questions or have a need for collaboration. My email is joshitr@missouri.edu, or you can find me on LinkedIn as well. And with that, I'll be happy to take any questions.
Thank you, Trupti. That was absolutely amazing to see how you brought all these things together and trying to make some sense out of it. We had discussed before that in the gene banks, we need a framework, we need a knowledge base where not only the scientists can look at the data, as you have shown, but we also want people like breeders and farmers and students and researchers and universities that may not have the knowledge to be able to distill some of that and select sessions from the gene bank in a better way than trying to the hit and miss way that they might be going about doing it right now. So this is an excellent presentation because it gives us the inspiration. If you did it for
Right.
multiple organisms, I think we can do it for the gene bank. So that was the purpose of, having you show us, maybe perhaps shine a light on the way. We have web pages, but they're not integrated just yet with all
Exactly.
the omics. We
Yeah.
have platforms which are good, but again, we lack the omics in it, and we lack the ability to be able to easily select things. So I think this was a really, really good introduction to all that. Thank you very much. We do have some questions from people. The first one is a very enthusiastic. Is it possible to organize hands-on workshop on soy KB?
Oh yeah, absolutely. And in fact, we have done several workshops in the past. So we actually have been regularly, I would say like every year, every 2 years, done a few workshops on soy KB at either the soybean breeders workshops or maybe even at larger conferences. So something like, PAG or other places. And so with our new soybean allele catalog, we are actually looking forward to come back and bring back with the workshops. It's just that in between with the COVID, we couldn't really hold some of them. But we have also done workshops as part of our NSF funded project where we also had the maize data and then we could have, we had KB Commons. So absolutely. I mean, if there's an interest, I'm happy to kind of even do a hands-on workshop. And that's what we have tried to do with all of our tools because that's really the best way to familiarize with the tool or get users involved in it because as you pointed earlier, that is the biggest challenge. We don't want to build something which only maybe a handful of computational or informatics folks really can follow or understand. Our goal is to reach to our breeders. Our goal is to reach to our farmers and researchers. And so we also involve them in the process when we are developing these tools in terms of what are you looking at? What is the piece we might be missing? What is the other information we need to bring in? So I am happy to, organize if there is an interest for Soy KB, KB Commons, any other tools that you might be wanting to look at. Sure.
Fantastic. Thank you. From Christelle, is there an interest or potential from KB Commons to collaborate and link with other types of public databases? For example, publicly accessible platforms
Yeah.
hosting data on plant genetic resources.
Yes, absolutely. And that's something that I did not get a chance to sort of mention. What we have done is we have built a lot of what is called as ETL pipelines, which is, extract, transform, and load information. So for example, as you know, GEO, fantastic resource for, say, gene expression, transcriptome, methylation, maybe other platforms exist where there's more proteomics data published resources. We have actually built this where we extract the information from there automatically and are able to bring it up into KB Commons. Now, I don't know if you're thinking more in terms of getting KB Commons piece because the main thing we need is just the connection, right? And so users can bring their own data if they want to just download one or two. But what we have built in the backend, I don't think it's come in the main development yet, is we actually tested this with a few organisms as to how efficiently can we pull out the data from time to time. So say something that we do on a quarterly basis, right? We pull up all the gene expression data and then load it into KB Commons so it becomes easily available for users. All you do is query with a keyword or maybe a dataset name, and then you already have that loaded in KB Commons. So we have always we have explored that, and we have also built a capability where we can handle fetching data automatically into KB Commons.
Wonderful, thanks. Um, our data, one of the questions, our data is often highly fragmented. Should
Mhm.
we invest on obtaining balanced data sets? Or what is the power of imputation to use such data?
Yeah.
And just to expand on that, we have data coming in from farmers, from people in other countries, researchers
Mhm.
that may or may not have a full grasp on how to
Yeah.
take the data.
Right.
How do you validate all that as you bring in that data?
Yes, I think that's an excellent question. And that is something that in the last 3 years, I think we spent a lot of time working on it as part of our soybean allele catalog development. As I mentioned, we did resequencing, right? So we had data which was looking quite complete for those 1,000 soybean accessions, but we also had interest in merging data sets that others generated. And so occasionally you will see there are positions where we don't have information for these other accessions, or there might be lines which are with a different coverage which give you a different number of SNPs. And so, in doing so, we actually tested two approaches. One approach was what we call as the fast merge, where we were doing and using a lot of imputation in between. So, all you're taking is mashing up two different data sets. And so, maybe a certain threshold that maybe, if it's less than 70% missing, then maybe you can work. But if it's 90% missing, you probably, it's too much. It's going to be bad quality imputation. And so we did that fast merge method that we think, which was more of an imputation approach. And then we also did the slow merge where we were actually calling all of the snips again for all these lines from raw data and merging with her, something that is a better curated panel. And so we did see that there's not a big difference between it. Of course, we decided to go later with the curated panel because now we are going even beyond 775 panel to about 2000. But the fast merge method actually did work well. And so I think from certain angles, it is okay to impute data as long as you are having at least a decent amount existing and you're not imputing the major chunk of it. But I think for a lot of tools and the allele diversity, for example, project that we are doing, imputation did work. And in fact, we are now working on adding features in that soybean allele, the second right hand component that I showed you, which will tell you now the pie chart of the phenotypic distribution as well. So you can decide which trait you want to query, and it'll tell you how many of those land races, sojas, and other what phenotypic distribution you have, and how many of those lines have an imputed or unimputated information. So we are actually bringing those pieces in, and I'm hoping it lands up going as part of the publication that we're working on. And so it's absolutely, I think, okay to do that. And we will have this toggle option. If you just purely want to use unimpute, you're okay using unimputed data, or you want, or okay using some amount of imitation, you can switch between it and also see how it impacts the results, perhaps online.
Thank you. Um, another from the same person. Banks often do not deal with single organisms, but with hundreds.
Mm-hmm.
How can we manage a knowledge base to encompass such diversity?
So,
How would we start? Where would we start by kind of creating something like that if we have a lot of diversity?
a lot of diversity. So essentially, you're looking something very similar to KB Commons framework, right? Where you have multiple organisms you're looking at, and even within an organism, you might have several references, right? Reference sequences or diverse lines that you want to track, and I mean, pan genome is also one thing that is on our radar. We still haven't gotten there or addressed that, but I think really it comes down to finding the commonalities between these organisms in terms of the computational framework as to how can you think what are the common pieces that you can start building the framework on, or what are going to be the common pieces that you have the most information available for. And then can you have modules where maybe for one organism you have data type A, but for the other organism you're going to miss that and perhaps have a data type B. So I think you kind of need that connectivity at the central, which is essentially how we approach KB Commons, right? You need that six essential files to get you started. Without that, I mean, there's a lot of organisms where you have unfinished genomes, or you may not have that beginning. So it becomes a little challenging because there's too many differences coming, right? But I think hopefully most of the organisms and crops you might be working with might still have that basic fundamental common piece. So starting up there and then definitely building infrastructure which is expandable, right? So that you don't have to every time keep making changes. I think that's the way we applied KB Commons, and we see the diversity of applications, right? Beyond crops, beyond biomedicine. In fact, I didn't mention, but we are working on a COVID surveillance, genomic surveillance project here in US, and that infrastructure has served us to even enable dashboards which can support that. So viruses, plants, diseases. I think it's possible to do all of that in one framework.
I like optimism. That's great. Uh, nice talk, Dr. Joshi, says somebody else. Am I correct to say that all of these new genomic or other omic tools help our efficiency to identify a genotype with one or more desirable traits, and plant breeders will still have to cross the desired types to develop cultivars which should be evaluated in multi-location replicated tests before releasing as a cultivar?
Yes. So you can always see that the computational and informatics, it will go up to a certain step, right? I don't think it's ever going to replace the validation pieces or maybe some of the crossing and, testing it in certain locations. You want to be absolutely sure before maybe you want to take it to the market or really make those decisions, right? But what you can see is instead of taking 20 years to get there, computational and informatics tools can apply a lot of these methods and help speed up the prediction, right? So that we can do a lot of these combinations. We can also generate more data if needed at times for the modeling, but really provide you with maybe top possible 4 or 5 candidates. And so what is really helping is it's cutting down this initial development piece, which I understand from a lot of our collaborators. Otherwise, the way is that you have to progress it, right, through years and different nurseries and so forth. And so you have to sort of think that it is a complementary approach. It is something that can make it more efficient for you, but I think there will still be a step where maybe we have a prediction with 90-95% confidence. You still want to go back and test it, right?
Yeah.
But at least you're not taking 10 and 20 years for that. Maybe this can happen in 2 or 3 years. So I think that way we can really expedite the process in that.
Yeah, yeah, very good. Next question. Very exciting tools. I'm really interested in using allele prediction and marker discovery.
Mhm.
Thanks for developing the tools. Are there any tools that integrate pangenomic data into the model in any of these that you have presented?
No, and I think that's what I mentioned about. So this is something that we are having quite a few discussions internally within our groups because a lot more, and there's more in terms of soybean right now because a lot of other genome reference genomes have become available, and we are looking at pangenome. It's easy to have each of these genomes separately, but I think what we are thinking is, whether how to incorporate that information into all of this allele diversity. And so I think this is something that is a question we still haven't solved in terms of bringing it back to the tools publicly right now. Internally, we have some ideas where we are looking at, perhaps if you're matching with something with B73, but then you have a lot of these other reference genomes, can we do a similar type of genomic variation calls on them and merge it? But however, some of these larger indels are going to be, pieces where, it's a lot easier to work with SNPs as compared to indels. And, for the soybean allele, we finished our 3-year United Soybean Board funding, and we have another year where we are actually focusing more on how to better curate these indels for these panels, and also look at some of the span genome questions and then promoter regions, intergenic regions for the traits. Because, we focused on gene allelic diversity initially, but there's also components and examples related to traits which come from these other genomic areas. And so that is work that we continue to do. And I hope maybe, we might be able to go in that direction.
This is a comment. It's a nice presentation and great integration of multi-state data. So just a little bit of a comment
Mhm.
there. The next one is, how would KB Commons interact with core collections and predictions of variants? And by core collections, they mean, if you distill out a very huge collection, you distill out the maximum diversity and you obtain, say, 10% or 1% if you want to make it mini core. So you have a
Right.
subset of
Subs-
the core,
yeah.
and that represents your maximum diversity. So the question is, how would the K Commons interact with those core collections and the predictions of the variations within and the diversity within?
Um, so I think and tell me if I got this kind of correct, but so for even in terms of KB Commons or Soy KB, we have this whole GRIN data set, so the 19,000 accessions. And then when we were doing our resequencing project, of course, the criteria there was at, the 10% diversity cutoff. And so we had selected lines that went into that. So if you're looking at that, maybe a larger data set exists and you want to automatically narrow it down based on diversity, I think it is possible for us to build tools which can help narrow that down. So either it can happen in terms of tool automatically on the framework, or the way we had that they were initially selected for resequencing, and that is the data that went into that. So I don't know if I answered the question or got the question correctly, but those are two approaches. And so we have also done such comparisons previously where we are looking at commonalities between SNP array data, which is really, really small subset, right? 50K versus our millions, and then really seeing which of these lines are showing the most similarity or diversity, and then pick some of them based on that. So I think there is room for incorporating such additional tools if there is a need to sort of, narrow it down further. In soybean allele catalog, when we did work, many a times some groups will filter out minor allele frequencies or whatever, 1%, 5%. For our work, because we were also interested in identifying and keeping some of these alleles, we kind of have kept all of that data. But it is possible to also maybe do some additional filtering based on that. Did I hope that answers maybe
Yes,
what
I
you're
think
looking
it did
for?
very well. Thanks.
Okay,
I apologize, we're a little bit over time, but if it's okay with you, Trupti, we have only one more question to
sure,
go.
sure,
And
not
it's
a problem.
always an interesting one. Where is the U.S. soybean gene bank and how do you interact with them?
So the U.S. soybean gene bank. So there is in Iowa, the USDA group has the USDA soybean database and kind of a gene bank, I believe, associated with that. We have interactions with the group whenever we are, say, publishing or making a lot of data available. For example, from our our PGen workflow, when we had data set for the initial 481 soybean lines, the whole SNP and Intel information we kind of shared with them. We actually were also linking back to them in terms of the gene information that they curate through our SoyKB website. And so what we have through our interactions with them, what we have seen is that we tend to work on complementary tools. And so many times their focus is more on providing information from the breeding standpoint or curated data sets for that. And what we have been focusing more on is computational and visualization pieces and prediction. And so that is where we see our complementarity exists, and wherever feasible we do the connections between them.
Thank you very much. Thanks again for a wonderful presentation, Trupti. This has inspired us to try and think of it in a
I'm
big
happy.
with CGIR and the Crop Trust thinking about a knowledge hub for
Yes,
everybody.
yes.
This has been quite inspirational. Thank you for your presentation today. That has
Yeah.
been.
Thank you for the
Thank you.
opportunity. I truly enjoyed it, and I'm happy if there are any follow-up questions or you have the need for getting into more details or any workshops. I'm happy to sort of follow up and have interactions with that.
Wonderful, wonderful. Thank you to Faye and Alyssa for your support today. Thank you to all the participants that have been here. We hope to see you next month for the last talk of the year. So until then, please stay safe and we'll see you soon. Bye-bye.
Bye bye, and thank you.
Bye, thank you.